Hello. So let's begin with another session of discussing MCQs which have been came in the NEET exam for last 38 years. So it is the second part of our lecture which I have been discussed in the last video live. So now let's begin with some very important questions. So the specialized cell for fixing atmospheric nitrogen in nostoc are. So it has come in NEET Karnataka 2013. So 4 options are given. You have to choose the correct answer for this question. So read the question first and then try to answer in the comment sections. Then I'll later tell you the answers with a proper explanation. So specialized cells for fixing atmospheric nitrogen in nostocs are echinates, heterocyst, hormogonia and nodules. So here the correct answer is heterocyst. So the explanation is the nostoc. A filamentous cyanobacteria and perform it can fix nitrogen from air by its specialized thick-walled cells, which is called heterocyst. These cells provide an anaerobic microenvironments by excluding oxygen and house the nitrogenase enzyme and enable conversion of nitrogen to ammonia. This is a classic adaptation in diazotrophic cyanobacteria, like cyanobacteria also called blue-green algae. And so the correct answer is heterocyst. And echinates dormant spore-like cells for survival, not nitrogen fixings. Hormogonia short filaments for reproduction and dispersal, and nodules root structures in legume for symbiotic nitrogen fixation with rhizobium, not in nostoc. So here the correct answer for this question is heterocyst. Let's move to the next question. So next question, the most abundant prokaryotes helpful to human in making curd from milk and in production of antibiotics are once categorized as option A cyanobacteria, option B archaebacteria, option C chemosynthetic autotrophs, and D heterotrophic bacteria. So here the correct answer will be heterotrophic bacteria. So heterotrophic bacteria which derive nutrition from organic compounds include key species like lactobacillus, lactose. So it ferments lactose in milk to produce lactic acid for curd production, and streptomyces or penicillium, though they are fungi, the bacterian analogous is called streptomyces for antibiotics like streptomycin. These are the most abundant and industrially exploited prokaryotes for these processes. So it is very helpful to humans. So the correct answer is heterotrophic bacteria will be the correct answer. And cyanobacteria mainly it is a photosynthetic nitrogen fixers not involved in any kind of curd or antibiotics production. So archaebacteria is a extremophile bacteria, rarely used in food or antibiotic productions. And chemosynthetic bacteria it gets its energy from chemical reactions not relevant to any dairy or pharma. So let's move to the next question for another question. The cyanobacteria are also referred to as option A protists, golden algae, slime molds, and blue-green algae. So the correct answer is the blue-green algae. Cyanobacteria also referred to as blue-green algae. Cyanobacteria one kind of prokaryotics. It is able to photosynthesis and it is commonly called blue-green algae for their pigmentations. They have chlorophyll A and phycobilins and it produces blue-green colors. and superficial resemblance to eukaryotic algae. That's why they are called blue-green algae. This traditional name persists in older literature despite their bacterial classification, distinguishing them from true algae. So true algae are mainly eukaryotic cells. So and blue-green algae are prokaryotic cells. So protists a diverse unicellular eukaryotic and not applicable to prokaryotic cyanobacteria. Golden algae refers to chrysophytes, eukaryotic algae with golden brown pigments, and slime molds eukaryotic protists with amoeboid and plasmoidal stage unrelated to cyanobacteria. So this question MCQ, the correct answer is the cyanobacteria is referred to as blue-green algae. Let's move to the next question. How many organisms in the list given below are autotrophs? So here lactobacillus, nostocs, cara, nitrosomonas, nitrobacter, streptomyces, saccharomyces, trypanosoma, porphyra, and ulfia. These organisms are given, and you have to find out which are among them are autotroph. That means they can produce their own food for survival. So let's flow. So the nostoc, cara, nitrosomonas, nitrobacter, porphyra, ulfia, that 6 are autotrophs. So and the others lactobacillus, streptomyces, saccharomyces, and trypanosoma are heterotrophs here. So the correct answer is 6. And nostoc, cara, nitrosomonas, nitrobacter, porphyra, ulfia. These 6 organisms are autotroph, and rest of them like lactobacillus, streptomyces, saccharomyces, trypanosoma are heterotrophs. Let's move to the next question. In eubacteria, a cellular component that resembles eukaryotic cells. So 4 options are given: plasma membrane, nucleus, ribosomes, and cell wall. So in eubacteria, a cellular component that resembles eukaryotes. So the ribosome will be the correct answer, because eubacterial ribosomes, mainly it is called 70S bacteria, are structurally and functionally similar to eukaryotic ribosomes. Both facilitate protein synthesis via mRNA translations. While smaller, they share core ribosomal RNA and operate analogously, reflecting evolutionary conservations. So, and the plasma membrane and nucleus and cell membranes are not resembles with bacteria. So, the eukaryotic plasma membrane, nucleus are and cell wall are totally different from eubacteria. So, let's move to the next question. Organisms called methanogens are most abundant in. So 4 options are given: sulfur rock, cattle yard, polluted stream, and 4 hot stream. So the correct answer for this question, the methanogens are mainly abundant in cattle yard, because methanogens are anaerobic archaea that produce methane via reduction of carbon dioxide or acetate. They thrive in oxygen-free, organic rich environment like ruminant guards, mainly in cow stomachs, making cattle yards is a hotspot. So this contributes about 25% of global methane emission from agriculture. So, and the other option like sulfur rock favors sulfur metabolizing archaea, not methanogens. Polluted stream, aerobic or facultative microbes dominate, and hot spring, thermophiles present, but methanogens are less abundant than in anaerobic digesters. So let's move to the next question. Bacterial leaf blight of rice is caused by a species of. 4 options are given: Xanthomonas, Pseudomonas, Alternaria, and Erwinia. So 4 options are given. So you have to find the correct answer from these options. So the correct answer will be the Xanthomonas. So bacterial leaf blight, a major rice disease causing wilting and yield loss caused by Xanthomonas oryzae. So that is the, and Pseudomonas. So the correct answer is Xanthomonas oryzae is a bacteria which cause the leaf blight of rice plant. So the Pseudomonas syringae cause bacterial brown spot in rice, not blight. And Alternaria, a fungal genus causing leaf spot in various crops, not bacterial blight. And Erwina cause soft roots in vegetables and fruit, not rice leaf blight. So here the other bacteria and their causes, their diseases also given. You can note down or take a screenshot from it. So you can note down or screenshot from this, and I'll discuss many more questions. So let me finish. I'm giving you some time that you can take the option. Okay. So yeah. So let's go to the next question. In the light of recent classification of living organisms into 3 domains of life, bacteria, archaea, and eukarya, which one of the following statement is true about archaea? So 4 options are given, 4 statements are given. You have to choose the true or correct statement. So archaea resembles eukarya in all aspects. Definitely not. Archaea have some novel features that are absent in other prokaryote and eukarya, and archaea completely differ from both prokaryote and eukarya, and archaea completely different from prokaryote. So here the correct statement will be the option B. The archaea have some novel features that are absent in other prokaryote as well as eukarya. So archaea form a distinct domain with unique traits like ether-linked isoprenoid membrane lipids, and which this type, and it is unique feature of archaea, ether-linked membrane lipids. But in bacteria they have ester-linked lipid membrane. So and histone-like protein for DNA packaging also present in archaea, but and in bacteria the histone protein like protein is totally absent, but in archaea the histone like protein and RNA polymerase resembling also present. So the histone protein is present in archaea and eukarya. That's why archaea share some characteristics with eukarya. And again another adaptation, these archaea enable able to adapt in extreme environment like a very high temperature, very high salty waters. So but bacteria lack this type of, most of the bacteria lack this type of adaptation, and bacteria also lack nucleus. So 3-domain system also in 3-domain system is proposed by UG in 1909, and which is based on 17S rRNA highlights these novelties. So resembles eukarya in all aspects. It is a false statement because they lack membrane bound organelles, archaeas, okay, and completely different from both prokaryote and eukaryotes. So overstate, they share prokaryotic simplicity with bacteria also, and completely different from prokaryote. It also false statement because archaea are prokaryotes differing biochemically from bacteria. That's why the correct answer is option B. Archaea have some novel features that are absent in some prokaryotes and eukarya. So you can note down this explanation, and it is a very good question. It came in NEET exam in 2008, and it have the probability to come in the next exam also. So let's move to the next question. Before you go to the next question, I give you 30 seconds. You take the screenshot. Let's move to the next question. So next question, Thermococcus, Methanococcus and Methanobacterium exemplify. So the four options are given. Archaebacteria that contain protein homologous to eukaryotic core histones. Archaebacteria that lack any histone resembling those found in eukaryotes but whose DNA is negatively supercoiled. Bacteria whose DNA is relaxed or positively supercoiled but which have cytoskeleton as well as mitochondria. And option D, bacteria that contain a skeleton and ribosome. So the correct answer for this question is option A, archaebacteria that contains protein homologous to eukaryotic core histone. So Thermococcus, Methanococcus and Methanobacteria are hyperthermic and methanogenic archaea that possess histone-like proteins homologous to the eukaryotic core histones. These proteins wrap DNA into nucleosome-like structure, aiding compaction and stability in extreme conditions, a feature unique to archaea among prokaryotes. Among Monera, mainly, this reflects their evolutionary link to the eukaryotes. So, archaea are evolutionary more resembles or more related to eukaryotes, not prokaryotes. So, and option B is not correct because it's lack histone but DNA negatively supercoiled. So archaea do have histones but negatively supercoiling is common in bacteria too. So that's why this is not the correct answer. Bacteria with relaxed or positive supercoiled DNA, cytoskeleton and mitochondria. These are archaea, not bacteria. Mitochondria are eukaryotic organelles. And bacteria with cytoskeleton and ribosomes. Archaea, not bacteria, have actin-like cytoskeleton proteins, but all prokaryotics have ribosomes. So, for this answer, it is a very critical type of question, and you have to be very careful when you answer this type of question. So, take the screenshot, and I'll go to the next question. So here for this question the correct answer is option B, archaebacteria that lack any histones resembling those found in eukaryotes but whose DNA negatively supercoiled. So let's move to the next question. So next question, which one of the following statement about mycoplasma is wrong? So option A, they are pleomorphic, they are sensitive to penicillin, they cause disease in plants, they are also called PPLO. So the correct answer for this question is they are sensitive to penicillin. So this is not true about mycoplasma because they are not sensitive to penicillin or any kind of antibiotics because they lack the cell wall, rendering them resistant to penicillin and other beta-lactam antibiotics which target cell wall synthesis. This is a key diagnostic trait leading to their use in minimal media without wall inhibitors. So pleomorphic means mycoplasma can change their shape. So as they don't have any kind of cell wall, that's why they don't have any kind of rigidity in their in their cell wall, so they can change their shape easily. And pleomorphic means they can change shape easily. And option C, cause disease in plants. Of course, phytoplasmas can cause disease like aster yellows and other plant diseases, and it is also called PPLO. It is also called PPLO. So mycoplasma are also called PPLO, pleuropneumonia-like organism. So because they discovered in the animal pleuropneumonia, so the mycoplasma are also called pleuropneumonia-like organism. Let's move to the next question. In which of the animals dimorphic nucleus is found? So 4 options are given. Very easy question. So Amoeba proteus, Trypanosoma gambians, Plasmodium vivax, and Paramecium. So the correct answer is here the Paramecium chordatum. They have the dimorphic nucleus for this question. So dimorphic nucleus refer to a cell with two types of nucleus, different in size and functions. One largest nucleus for metabolism and small nucleus is reproduction. So Amoeba proteus has a single monomorphic nucleus, and Plasmodium vivax, it is a malaria parasites with a single nucleus. So the option, the Paramecium chordatum have a two different type of nucleus. So that's why the correct answer is here Paramecium chordatum. Let's move to the next question. The most thoroughly studied fact of the known bacteria plant interaction is the cyanobacteria symbiosis with some aquatic ferns, gall formation on certain angiosperms by agrobacterium, nodulation of Sesbania system by nitrogen fixing bacteria, plant growth stimulation by phosphate solubilizing bacteria. So the correct answer is option B, gall formation on certain angiosperm bacteria. So agrobacterium tumefaciens induce gall disease, crown gall disease, by transferring its tDNA into the plant cells. So the correct answer is option B, gall formation on certain angiosperms by agrobacterium bacterium. So let's move to the next question. The chief advantage of encysment to an amoeba is. So 4 options are given. Read the 4 options and try to answer. So first option is the chance to get rid of accumulated waste product, the ability to survive during adverse physical condition, the ability to live for some time without ingesting food, protection from parasites and predators. So the correct answer is the option amoeba mainly form encysment around them when they need to survive in adverse physical condition. So the correct answer is the ability to survive during adverse condition is the correct answer. So encysment in amoeba involves forming a protective cyst wall, mainly from glycoproteins around the cell, allowing it to withstand desiccation, extreme temperatures, and nutrient scarcity. This dominant stage enables survival and dispersal until the condition improves, after which encystment occurs. It is a key adaptation in free-living protozoans as well as. So protozoan is amoeba. So amoeba. Let's move to the next question. So the get rid of waste. So get rid of waste product. So waste product is waste is expelled via contractile vacuoles, not encystment. So live without food. It also causes the encystment, but it is a secondary. So survival is the main primary benefits the amoeba got. So that's why the C option is not correct. And protection from parasites or predator, it also beneficial, partially benefited, but it is not the main primary cause of enlistment is the surviving the adverse conditions. So let's move to the next question. So next question, which bacteria is utilized in gobar gas plant? So gobar gas plants, methanogens, nitrifying bacteria, ammonifying bacteria, and nitrifying bacteria. So the correct answer is methanogens. Definitely gobar gas in gobar, cow dung. Gobar means here cow dung plant. Like in village type of in village, many people create one type of plants where the gobar or cow dung. Gobar means here cow dung is used to produce gas for fuel, like in cooking, lighting in household. That's why here we know that in cow's stomach or ruminant stomachs, the methanogens archaea bacteria are present and they can produce methane gas. That's why the methanogens will be the correct answer for this question. So other options are not correct because the nitrifying bacteria mainly aerobic oxidizer and it can convert ammonia to nitrite or nitrates, and ammonifying bacteria decompose organic into ammonia, and denitrifying bacteria it also convert nitrate to nitrogen, so it not produce methane gas. That's why the correct answer is methanogen. So let's move to the next question. Which statement is correct for bacterial transduction? So 4 options are given. You have to choose the correct answer. Transfer of some genes from one bacteria to another bacteria. Transfer of genes by conjugation bacteria to another. Bacteria obtains its DNA directly. Bacteria obtains its DNA from other external sources. So the correct answer is here transfer of some genes from one bacteria to another bacteria through virus. So bacterial transduction is a horizontal gene transfer from so it is the correct answer for this question is option B. Let's move to the next, and the other option conjugation direct cell to cell transfer via sex peleas not involving virus. Direct DNA refers to the transformation uptake of naked DNA, not transduction. External source vague but implies transformation, not virus mediated. Let's move to the next question. What is true for cyanobacteria? So 4 options are oxygenic with nitrogenase, oxygenic without nitrogenase, and non-oxygenic with nitrogenase, and non-oxygenic without nitrogenase. So the correct answer is oxygenic with nitrogenase. So cyanobacteria are oxygenic phototrophs which can perform photosynthesis producing oxygen via photosynthesis, and many possess nitrogen enzyme in specialized cell which is called heterocyst for nitrogen fixation. Let's move to the next question. What is the true for archaebacteria? All halophiles, all photosynthetic, all fossils, and option D, oldest living beings. So the correct answer is here option D, they are oldest living beings. So archaebacteria represent the oldest extant, mean it is living domain of life with molecular clocks. Mainly it originated 3.8 billion years ago. So the oldest living organism here is the archaebacteria. Let's move to the next question. The main difference in between gram positive and gram negative bacteria resides in their cell wall. Option B, cell membrane. Option C, cytoplasm. Option D, flagella. So the correct answer is cell wall. The gram positive bacteria have a thick polypeptidoglycan layer in their cell wall retaining crestal violet stain, but in gram-negative bacteria have a thin peptidoglycan layer and outer lipopolysaccharides, and it's loose. It cannot hold the gram crystal violet stains. That's why they are called gram-negative. So the main difference between main difference between gram positive and gram negative bacteria resides in their cell wall. So the correct answer is here is cell wall. Let's move to the next question. Photosynthetic bacteria have pigments in leucoplast, chloroplast, chromoplast, chromatophores. So the correct answer is chromoplast. So photosynthetic bacteria, mainly purple null sulfur bacteria, use chromatophore intracytoplasmic membrane invagination containing bacteriochlorophyll and carotenoids for anoxygenic photosynthesis. These vesicles harvest light without producing oxygen, unlike eukaryotic chloroplast. So the chromoplast will be the correct answer. So photosynthetic bacteria have pigments, not leucoplast, not chloroplast, not chromoplast. So the chromatophores. So the chromatophores will be the correct answer for this question. Let's move to the next question. In prokaryotes, the genetic material is option A, linear DNA with histone, circular DNA with histone, linear DNA without histone, circular DNA without histone. So prokaryotic genetic material is a circular DNA without histone. So the correct answer will be the circular DNA without histones. Prokaryotic bacteria have a single circular chromosome in nucleide, not packaged with histones. So that's why the prokaryotics, their genetic material is circular DNA without histones. So the prokaryotic bacteria prokaryotes don't have any kind of histones. Let's move to the next question. A few organisms are known to grow and multiply at temperature of 100 to 105 degree centigrade. They belong to marine archaebacteria, thermophilic sulfur bacteria, hot spring blue-green algae, thermophilic subaerial fungi. So the correct answer for this question is thermophilic sulfur bacteria. So thermophilic sulfur bacteria, they are very tolerable to the high temperature. Also, it lives in acidic hot spring. They represent archaea adapted to extreme heat because they have heat stable enzymes and membrane. So let's move to the next question. So next questions are from the Protista. Which of the following organisms are known as chief producer in ocean? So the four options are given: dinoflagellates, diatoms, euglenoids, and cyanobacteria. So for this question, I will discuss in the next live. Today I will finish this session for the day. So I'm finishing.